Hello students, let's talk about principles of biotechnology and the various nuances of biotechnology including genetic engineering and all. So I'll start with the basic fundamental definition of the biotechnology. Before jumping into the different steps in the processes of biotechnology, we need to understand this fact that in this 20th century, uh, it is a biology among the scientific subjects, or apart from physics and chemistry, whatever the revolutionary aspect that has been happened in the 20th century has opened up the window for the plethora of manipulations that can be done just for the sake of our anthropogenic use. That means the humans have advanced the technology and they have utilized the other part of the science like physics and chemistry and biology, that we are able to manipulate so much even the organisms. So biotechnology is something into which we try to manipulate the organisms or their organelles or their parts just for the sake of the development of the unique products for the human use. So you see, biotechnology was, this word was coined and invented by Carl Irke. Carl Irke. He discovered this term of biotechnology and approximately 50 years back, and he was a Hungarian scientist, and with the help of the biotechnology, he manipulated the genes among the pigs, and he able to produce a lot of pigs that was genetically modified just for the sake of human use. So you see, biotechnology as a term can be defined by the European Federation of Biotechnology. This EFB body is it's basically an integration of the traditional science with the organisms and cells and thereof parts of the organisms, basically, and whether it is a cellular part or the molecular part, just for the sake of the development of a lot of different biotechnological products. Just like antibiotics, you see number of cells can be manipulated and the genes can be manipulated and developed in the form of the DNA vaccines. So lot many things that can be developed and manipulated in terms of the molecular analogous substance which is present in our cells. So biotechnology is something which is an integrated part of the traditional biological science with the molecular biology through which we can able to produce a lot of different products just for the anthropogenic use, and that products can be produced on the larger basis, on the larger scale. So let's now try to understand what is biotechnology and what are different steps of biotechnology. There are two different aims, or you can see the techniques that is basically been utilized or performed in the biotechnology. The first technique is just to create a sterile environment, just to create sterile environment in microbes so that the microbes can be cultured out and we can produce microbes on the larger scale just for able to synthesis of a lot of different products which is utilized by humans, like ethanol production and antibiotic production. Ethanol production is basically performed by larger scale on the basis of yeast. So yeast has been utilized and it has been cultured on the larger scale basis, and in order to produce ethanol or the yeast on the larger scale, what you have to do is to create a sterile environment so that the other organism cannot infect in this particular a sterile environment, wherever in this particular chamber this organism which has been manipulated on the basis to produce the ethanol on the larger basis with the help of the process called as fermentation. This is some of the instances where the biotechnology word has been used. Now the second important technique that biotechnology used is a genetic engineering. Genetic engineering is is one of the venture which has been given by biotechnology, into which the organism or there is a manipulation in their chemical structure of the DNA or the RNA which is done, and that leads to the production of some different type of organism that would having the desirable genes or desirable DNA, or ultimately it will produce the desirable quality that we want to have. So with the help of genetic engineering, we can produce certain microorganism, or even we can manipulate the DNA or RNA of a particular organism, and we can insert or we can remove certain genes which is been desirable for us, and we can produce that gene on the larger scale, which is very much important for the human. So genetic engineering is also called as rDNA technology. rDNA technology. That means the recombinant DNA technology. So you can see there are the two principles or the techniques in the biotechnology which has been utilized by and large, is to create a sterile environment just for the sake of the production on the larger scale basis for either the microorganism or a different type of organism which is able to produce, the desirable product on the larger scale. And the second thing is the genetic engineering, which itself is a manipulation in the genetic material of an organism that leads to the production of the genes, or on the larger scale basis that is having the desirable DNA insertion. So let's talk about the genetic engineering or the recombinant DNA technology, which is one of the most important part of the biotechnology in the larger basis. So what is genetic engineering or rDNA technology or recombinant DNA technology? There are number of processes or steps that we see that we utilize in rDNA technology that we'll understand later on. But before that, the basic principle behind genetic engineering or biotechnology or genetic engineering or rDNA technology that we need to understand, as we see that sexual reproduction provides us the opportunity to give the variation, right? So variation comes from the sexual reproduction because the recombination phenomena happens during the meiotic division, and you see after the fertilization, when the zygote formation happens, the half of the chromosomes comes from the each parent, and they comes together and they form a zygote, and that zygote, since have the half of the genetic material from the each parent, there is a scope of recombination between all these genes which is coming from the each parent, and then ultimately we have the variation. But although on the other hand, in asexual reproduction it does not happen, it just carries their parental genes. So you see, one of the adversity or the adverse effect of sexual reproduction or the asexual reproduction is that the genes which are not desirable or which are very much not effective in the due course of the evolution, which has also been carried forward with the help of the sexual reproduction as well as the asexual reproduction. Just to come over of this kind of situation, we have this artificial venture or the artificial platform that is called as the recombinant DNA technology, through which we can, if we take out that the faulty gene which is present in the organism, and if we insert the genes which is desirable to us, which is having the quality that we want, if we can insert that gene back into the organism, definitely the type of product or we are expecting in the organism that can be restored. So this is one of the fundamental aspects behind this genetic engineering, that why do we do this manipulation in the DNA or RNA just to have our desired quality in a particular organism. So let's talk about the steps of genetic engineering or rDNA tech. What are the steps? The first step is the isolation of DNA, and desirable gene second thing is just to integrate the gene with suitable into suitable vector. And third is transformation of recombinant gene. That integration of this gene into suitable vector gives rise to the recombinant gene. And finally, the transformation of the recombinant gene into host species leads to desirable product. So these are some fundamental steps. The first step is isolation of DNA and the desirable gene. Isolation of DNA means what? Isolation of DNA from certain kind of plasmid or the vector that we'll talk about later on, and the desirable gene that we are wanting to incorporate into the vector. So let's talk about this first step of isolation of DNA and desirable gene. This isolation of DNA can be basically isolated from the organism, that the DNA that we are wanting to manipulate with. So suppose the DNA that we would like to manipulate with, we have to first isolate it. There are the different method of isolation through which we can isolate the DNA and we can fragment it. So the first step is isolation of DNA and then fragment or cut it, restriction endonucleases. They are the different type of enzymes which has been discovered in the bacterial system that has a very strong capacity to recognize as well as cut to the certain specific portion of the DNA which is present into the either into the bacteria or it is present into the host organism. So the two type of DNA here we are wanting: one DNA which is coming from the organism that we would like to manipulate it, and the second DNA which is coming from this vector or plasmid. Let me explain you what is vector. Vector is something that it is a kind of DNA that has the ability to take up, or it has the ability to incorporate the foreign DNA molecule ultimately with the help of the particular enzyme called as DNA restriction endonucleases, as well as the DNA ligase. Later on I will explain that how this DNA ligase has the ability to paste that DNA molecule from the foreign origin and with the vector molecule, and ultimately it has ability to replicate into the into the bacteria basically, and the ability to replication of that particular foreign inserted DNA molecule leads to the production, or it is the amplification of that particular DNA molecule present into the bacteria. Let's say the best example of this vector is the plasmid. There are number of plasmids which is present into the bacterial system, and these plasmids are nothing but the extra chromosomal material which has the ability to replicate, and that also contains number of sites in their DNA which gives them the ability to bypass or to grow into certain kind of antibiotics. So it provides certain kind of antibiotic resistance. So you see, as far as the examples are concerned, there are the different type of plasmids present in the bacterial system which has extra chromosomal material, and these extra chromosomal material are not governed by basically, they are entirely independent from the bacterial own genetic material. So that is the beauty of having this plasmid. There are the number of plasmid like F1 plasmid, R plasmid, or CF plasmid, R plasmid, they are called plasmid. These type of plasmids provides the extra edge or the quality to the bacteria, that the bacteria does not have that kind of quality present in their own genetic material. As for example, this F plasmid provides the bacteria to do the conjugation into the adverse condition, right, where with the help of F plasmid, it can produce the sex pili. So R plasmid is providing the bacteria to the edge where it can grow up in itself into certain kind of antibiotic. So it provides the antibiotic resistance. The col plasmid is a type of plasmid where it provides the bacteria to cope up with the environment where bacteriophage acted on this particular bacteria. So you just get rid of from the bacteriophages, because you know that the bacteriophage can eat up the bacteria. So this kind of plasmid always help the bacteria to produce certain kind of quality, so that the bacteria can live or survive in the particular environment in a better way or other. So plasmids are very important and the wonderful molecules which is present into the bacteria, and it replicates independently from the bacterial genetic system. So you see that bacterial extra chromosomes or the plasmids have, suppose this is the plasmid, the example of plasmid is PBR322. If you go through the genetic material or the DNA of the plasmid, it has certain kind of different sites that provide certain kind of qualities. Like suppose this site provides the recognition site for the E. coli. E. coli is one of the type of restriction endonucleases that has the ability to recognize a certain sequence at the particular site. So this is the E. coli site. There is a certain site that provides for ampicillin resistance. Ampicillin resistance. And there are certain sites that has ability to provide, tetracycline resistance. So you see, that means if the bacteria is growing into the environment where certain kind of antibiotics are there, the bacteria which will be having this kind of plasmid will be able to survive. Other bacterial system will not be able to survive because they don't have such kind of plasmid system where the genes are present that provides the bacteria for the antibiotic resistance. So there are two different sites you see in the PBR322, the ampicillin resistant genes are there and tetracycline resistance genes are there. And the one site that provides this EcoR1. So now come to the second point, that how do we fragment or cut this this DNA molecule which is coming from the organism, and it is coming from the plasmid. So you see that the important enzyme are restriction endonucleases. There are two type of endonucleases. One is the exonucleases, second is the endonucleases. Exonucleases are the type of enzymes that cut the DNA at their terminal position, but the endonucleases are the type of enzymes that cut the DNA portion somewhere in between. So restriction endonucleases is one of the type of endonucleases that can be of three different type, RE1, RE2 and RE3. So the first restriction endonucleases that was discovered was named as HEN2. It was the first restriction endonucleases discovered in 1972. So you see that the function of restriction endonucleases are very specific because they always recognize certain sequence present in the vector or into the other the DNA sequences, where the DNA sequences are palindromic in nature. Palindromes. They always recognize the palindromes. What are palindromes? Palindromes are those DNA sequence which can be which has a similar type of repeat. You start reading from the one end or from the another end. As for example, AGCT. You see AGCT. You see, suppose this is the DNA sequence, that you can read this DNA sequence from left to right. You see AGCT, that is the base, and if you read it from this side, that will find that exactly the sequence is exactly same, that is at the opposite end. That means if you can read something, it is just like reading some word like a Malayalam. If you read this word from this side, Malayalam, and from this side, Malayalam, both will be exactly similar. So this kind of word or the sentences which can be written or can be read exactly from their one to the opposite end, such a phenomena is called as a palindrome. So there are certain sequences present in the DNA, whether it is the plasmid DNA or it is in the host DNA or in the DNA of the organism, where restriction endonucleases can recognize this particular site of palindromic sequence, and exactly it will recognize it and cut it in both the DNAs, so that a sticky site can be created. So the way it cuts, it always create a sticky site. What kind of sticky site it creates? So suppose, as for example, you take the HIN2, HIND2. HIN2 is a kind of restriction endonucleases. It cuts like, it will produce the nick here, and it will put the nick here, so that what will be produced is A, C, C, G is a 1, and the other will be G, C, T, A. Do you see that? You see the nick is there at this particular position and at this particular position. So there are two type of ends which has been created. One end is the overhang here. This is the one end which is overhang like this. So A is overhang, and the second is overhanging like this. You see two different type of sequence that has an overhang at the terminal position. So such kind of overhang is been created by restriction endonucleases, so that this end provides the sticky end. So that if the foreign DNA molecule can come and incorporate and exactly go stick to this particular position and this particular position with the help of the DNA ligase. DNA ligase is a very important enzyme that helps into the sticking, or it helps into the incorporation of the foreign DNA molecule into the fragmented DNA molecule cut by the restriction endonucleases. So you see how we cut these two different type of DNA molecules is with the help of restriction endonucleases. Now the question arises, once we have a lot of different fragmentation occurs in the DNA sequence which has been isolated from the organism, that we have to manipulate it. How do we recognize it? We recognize it with the help of the technique called as gel electrophoresis. Agarose gel electrophoresis. Agarose gel electrophoresis is based upon certain kind of electricity field, electricity based technique, where we provide both the type of charges, positive charge as well as the negative charge. As you understand, is the particular fact that the DNA is a negatively charged molecule. So if it will provide this kind of DNA, different fragmented DNA molecule into the electrophoretic system, where the positive and the negative pole has been created with the help of the cathode and anode, definitely if we'll put up the DNA into this kind of system which is having the agarose, DNA will always migrate to the positive charge. That means it will always migrate to the anode. So it is by compulsion that the different fragmented DNA molecule will be migrated from the negative pole to the positive pole, that means from the cathode to anode in a particular plate, electroplate, where it has been charged. So if this is the this is the anode, where this DNA molecule has been migrating, and it will be migrated according to their molecular weight or according to their size, since it is migrating into a gel which is made upon agar. You know that the agar is a kind of gelatinous substance which provides certain kind of matrix or certain kind of platform which is having number of pore size. So it is having the differential pore size through which DNA according to their size can percolate, and it can migrate from one position to the another position. So smaller the fragment of the DNA, it has better the ability to migrate from one position to another position. So you'll find out at the end of this particular pole, the smaller fragment will be separated. It is a separation technique. So this separation technique is based upon the electricity through which we separate the fragmented DNA molecule which has been fragmented by number of restriction endonucleases like EcoR1, HIND, HIN2, and there are different type of restriction endonucleases. And then what do that once we have an idea that these DNA fragmented molecule has been separated, or it has been clearly seen on the basis of their weight as well as the size, it has been migrated from the negative to the positive pole. The next aim is to just isolate that particular DNA molecule or recognize the DNA of our interest, because the DNA of our interest is basically mixed up in these fragmented DNA parts or the molecule. So how do we recognize our DNA of interest from this junk? We utilize the light called as UV light, and we stain this entire DNA fragmented molecule with the help of ethidium bromide, ETBr. ETBr is one of the most important type of chemical that has the ability to stain the nucleic acid, whether it is a DNA or the RNA molecule. So once it has been stained in the ETBr, and then if you insert the plate into the UV rays, then these bands are clearly been seen with the different type of color. The orange color bands has been basically seen because this ETBr so plate has been recognized by the UV light, and on the basis of that, a certain fragment that we can identify that has a certain base pair or certain length, that has been recognized with the help of the probe molecule that you have already been put up before inserting into the UV light. So with this, once we have the idea about our fragmented molecule, the DNA of our interest, then the next step would be to insert that DNA molecule into the vector molecule with the help of a DNA ligase, because we cut the same restriction endonucleases with the host with the DNA of our interest as well as the vector molecular DNA, and both the cut will be exactly similar, and then both the foreign DNA can be inserted into the plasmid DNA or the vector DNA with the help of the DNA ligase. Once this has been incorporated, this DNA is basically called as recombinant DNA. So you see that suppose this was our DNA of interest that was inserted into this foreign DNA, and this is basically vector DNA. This insertion was made with the help of a specific restriction endonuclease molecule. Once it has been made, this insertion is completed. This is called as recombinant DNA. Now this DNA will be then transformed into the host. So the step is called transformation. Or transformation is another important event where you have to find a suitable host where this DNA molecule, this recombinant DNA molecule can be inserted, and where the replication of this DNA molecule should takes place, and ultimately the amplified DNA, the number of different recombinant DNA can be produced into the larger scale. So how do we achieve this thing? That we just insert it into the certain bacteria that has this kind of plasmid DNA that is already there. So once you insert, find out the suitable host which is related to the bacteria through which this plasmid was isolated. So you can insert this recombinant DNA and you just culture it for some point of time, and then after culturing this into a certain media, you'll find out after certain time later this recombinant DNA can be multiplied or replicated various number of times, and the different number of plasmid has been come out with that is having this foreign DNA molecule. So with the help of this, you can amplify it or you can multiply the number of this inserted foreign DNA molecules, and ultimately with the help of this DNA molecule, ultimately the protein has been created or formed, that because of the translation process where it participates, ultimately you take out the protein that you are desired because of the desired gene you have inserted into the bacteria. So you see, these three events are very important part of the recombinant DNA technology, and with the help of that, the different type of manipulations were done not only in the plants but also in the animals. As far as you see this kind of technology is utilized in the case of plant, we'll take up the example of the agrobacterium. You know that Agrobacterium tumefaciens is one of the pathogenic bacteria that create a lot of different diseases in the plants. One of the diseases is called crown gall disease. So what happens? Agrobacterium generally infects the plant, and it releases different type of chemicals that leads to the the tumor-like structure that is created in the plant. But if you utilize this agrobacterium by inserting the foreign DNA molecule, that has the ability to stop this tumor formation with the help of this Ti plasmid, which gives this edge to the bacteria, that if you manipulate this Ti plasmid by inserting the foreign DNA molecule that is desirable, this Ti plasmid through the agrobacterium will not able to create the crown gall disease, but will be able to create the protein which is desirable to us. So you see, these vectors and plasmids and bacteria can be a very special tool for us, which can utilize as a vehicle, and ultimately it has been producing a lot of different chemicals and substance which is desirable to us. With the help of this genetic engineering, a lot of different transgenic animals and plants has been made that gives us a lot of different desirable quality that we wanted. And as far as the animals are concerned, different type of cloning has been performed, you see, in the case of animals. The first time the cloning was performed in the animal was in 1990s or in 1991, where the Dolly named ship was, first time a ship was been basically cloned, and the name of that ship was Dolly. The cells that diploid cells that was isolated from the udder of this ship, the mother, basically from the mammary gland, and it was enucleated basically, and their genetic material was been manipulated by giving the desirable gene. So without the father, you see, Dolly was born because the cell was manipulated, was enucleated, and and injected the nucleus that is carrying the genetic material which is already deployed, that has the ability to, that has the ability to gives ability to differentiate as well as the replicate, and then ultimately a new individual is formed in the form of clone. Why the word clone was utilized? Because this dolly was exactly similar to her mother. So you see, with the help of genetic engineering, a lot of different anthropogenic animals as well as the plants can be created, and that can be one of the boon and a certain kind of ethical issues, social issues are there in genetic engineering, where you see certain kind of experiments which are inhibited, which are not promoted, and which is basically not authorized. As you see, the cloning of the humans are not authorized. So that is the important aspect of the biotechnology and the rDNA technology, which is acting as a very strong tool, not only in the modern biological science, but in the future also we can have a better idea that how do we manipulate the gene so that we cannot have the disease which is not desirable for us in the future. So with this, a lot of different therapeutic approach and the vaccines has been created, which is based upon this molecular approach, where the DNA vaccines can be created, which is customized to a particular person that has a certain kind of genetic disorder or certain kind of disability which is genetic in nature. So that is all about the genetic engineering and their steps and the principles of biotechnology. Thank you.